All right, it is noon, Friday, October 9th, 2020. I want to welcome everybody back to the Medical Biochemical Genetics Clinical Course Seminar Series. Today we have the pleasure of listening to Dr. Austin Larson talk about congenital disorders of glycosylation and an introduction into the clinical and phenotypic aspects of mitochondrial disorders. We'll be hearing a little bit more about mitochondrial disorders in a few weeks from Johan van Hove. It'll be more laboratory aspects and potential therapies. And next week, we'll be welcoming Dr. Charles Venditti to talk about methylmalonic acidemia, propionic acidemia, and other organic acidemias if there's time. As always, if there are questions, please hold them to the end. Make sure you're muted during the lecture, and then afterwards you can unmute to ask your question if you need to. If you could direct them to me in the chat, I can help sort of funnel things to Austin for answering. All other housekeeping things, so the lecture should be available in my Dropbox, but also we've graciously been able to have SIMD carry the lectures on their website. So they're archived now on the SIMD website, available to everyone with the link, and Holly can send out that link after the lecture if you don't already have it. And again, spread the word, let people know if they want to get on and join and be part of these lectures, they can. We go through November 20th, and then we will figure out what we're going to be doing for the remainder of the academic year and try to restart again in July with some improvements, hopefully. So without further ado, I will give this over to Austin and go for it. Thank you.
Thank you, Pete. So thank you all for being here and listening. This will be sort of by necessity a pretty superficial look at two very large topics. So my hope today is to kind of give you the lay of the land and give you the context that will help you to dig into the research and sort of do your own research going forward when you have patients with specific CDGs or mitochondrial diseases. As Pete said, the mitochondrial piece of this is really meant to be the kind of phenotypes and genetics with Johan van Hove going much more deeply into the biochemistry, the mechanisms and potential therapies for mitochondrial disease in a couple weeks. So CDGs are a very large group of disorders. This is starting to be a relatively old slide, but it demonstrates the point that as of 2013, there were over 100 different discrete genetic CDGs that had been identified. You can see kind of a pattern in the specific types of CDGs that were identified and when they were identified. So those CDGs that have a really clear biochemical signature or really obvious findings on histopathology were identified earlier on. So those include the N-linked disorders of glycosylation that can often be identified biochemically, the dystroglycanopathies that can be identified based on immunohistochemistry and other pathology studies. And then later on, you can see things like a lot of the GPI anchor disorders being identified because those really require broad genetic testing and have somewhat non-specific phenotypes and no really clear biochemical marker. So I'm going to talk about most of these pathways and try to give you at least one example of each of these types of CDGs. So to start with, the process of glycosylation is a post-translational modification of a protein. The proteins that are modified with glycosylation are usually either cell surface or secreted proteins. And there are some lipids that we'll talk about that go through a process of glycosylation. And the purpose of glycosylating them is to change their properties. So to make them fold differently, to give them different charges, to give them different spatial characteristics, sometimes to give them a specific recognizable identity. So that's really relevant in the immune system to have cell surface identifiers that are often glycans. And there are a lot of different glycosylation pathways. So I'm going to go through some vocabulary here. So there's an alphabet of sugars that is used to represent different glycan structures. And that alphabet is used in every publication and every representation of glycans. So for example, mannose, which will come up a lot, mannose is always represented by a green circle. Galactose is always represented by a yellow circle. Fucose by a red triangle. So that alphabet is used consistently, and you can look for that. Hopefully there will usually be a legend, but if not, you'll know that, that those are consistent representations of those sugars. And so you can see that those glycan structures have multiple sugars that are linked together. The link to the protein can either be an N link, which is a nitrogen bond, or an O link, which is an oxygen bond to the protein in question. And so those N-linked bonds, those nitrogen links, can happen on an asparagine amino acid. The O-linked bonds can happen on either a serine or a threonine. So there are only 3 amino acids that can be glycosylated in order to modify proteins. And so if you're thinking about a potential missense change in a gene and whether that's physiologically relevant, one thing to think about if it's an asparagine, a serine, or a threonine is whether that's a glycosylation site, in which case it may be a particularly relevant missense change. Within the cell, glycosylation occurs in the endoplasmic reticulum and in the Golgi. So we'll talk about some disruptions of those organelles and how those disruptions can result in sort of a secondary disorder of glycosylation, meaning not a single enzyme like a specific transferase enzyme, but a more global process that impacts those organelles that are responsible for glycosylation and hence has kind of diffuse dysregulation of glycosylation as a result. So those N-linked glycans can be quite complicated. There are a couple different subtypes. So there are what's called high mannose N-linked glycans. Those have primarily mannose sugars that are attached to one another. There are complex glycan structures, kind of multiple branches. And one of the things that's often true about those complex N-linked glycan structures is that they may have a terminal sialic acid. And that's important because that sialic acid has a negative charge. And the number of sialic acid residues gives more negative charge to that glycosylated protein, and hence changes the characteristics or the way that protein behaves. And that charged sialic acid can also give us a really good diagnostic tool, and we'll get into that. There are a couple different O-linked glycosylation pathways, and a number of the pathways are referred to by the first sugar that is attached to the protein. So by that O-link. So the O-linked mannosylation is a pathway that broadly leads to the group of disorders that we call dystroglycanopathies. O-xylosylation is so xylose is the first O-linked sugar in the glycan structure in mucopolysaccharide synthesis. So O-linked xylosylation defects lead to mucopolysaccharide synthesis disorders. We'll also talk about a group of disorders called GPI anchor disorders. So this GPI anchor, you can see kind of in the bottom middle of this representation. The phosphatidylinositol is a lipid species that embeds itself in the membrane. And then the glycosyl piece of that GPI anchor is the link between that lipid species and a protein that is bound to the cell surface. So the GPI anchor disorders lead to a disruption of those proteins that are bound to the cell surface. Glycosphingolipids are obviously not proteins, but lipids that often have a really important neurologic role, and hence those are a group of primarily neurologic disorders that we'll talk about. And then the last thing that I want to mention in terms of our basic vocabulary is this molecule called dolicol. So dolicol is a molecule that's made as part of the sort of cholesterol synthesis, related to cholesterol synthesis. And dolicol is another lipid that can be embedded in a membrane, in this case, the membrane of the endoplasmic reticulum or the Golgi, and that can hold that, that protein in place in the ER and the Golgi while it undergoes glycosylation. So disorders of dolicol synthesis can lead to disruptions of multiple glycosylation pathways. So the traditional diagnostic technique that was used to identify disorders of glycosylation was called transferrin isoelectric focusing. So transferrin is a protein that probably everyone is familiar with. It's involved in iron transport, and it's a secreted protein, so it's present in the plasma. And it has this very characteristic pattern of glycosylation where it has two sites for N-linked glycosylation. The two glycans that are attached at those sites are identical to each other, and they both have terminal sialic acid residues. As I mentioned earlier, those sialic acid residues have a negative charge. So one of the ways that you could identify a disorder of glycosylation is to look at the kind of charge to mass ratio of transferrin and look at the banding pattern that results when you run that protein on a gel. So the more sialic acid residues that are attached to that transferrin, the more it migrates on the gel because it has a stronger charge. So a control transferrin protein would migrate about this far, and you get a big band where all of the proteins end up that have 4 sialic acid residues. Now, when there are disorders of glycosylation, there are kind of 2 categories of dysfunction. So there's a kind of dysfunction where you just can't make as many glycans. And so in that case, the glycans that you do make have a normal formation, but you just don't have as many of them. So in that scenario, you had a big band at 2 sialic acids and a big band at 4 sialic acids. So that was termed a type 1 CDG. And that's in contrast to the type 2 CDGs, where those are sort of later in the pathway of N-glycan synthesis and modification. And in that case, you don't just end up with 0, 2, or 4 sialic acid residues, you would end up with 1, 2, 3, or 4. So this is termed a type 2 CDG pattern. So this is kind of the classic diagnostic technique that was used. As I said, the type 1 and type 2 patterns that you see sort of segregate by where in the pathway of N-glycan synthesis the problem is occurring. So these early defects, like for example, PMM2-CDG, which we'll talk about in detail, means that you sort of just lack the substrate to make these glycan structures. And as a result, you just don't make as many of them. In contrast, these late defects, these enzymes that are more active in the Golgi or sort of more global dysfunction of the Golgi, that leads to abnormal modification of the glycans that are created. And so that's that type 2 pattern. And so for a long time, the CDGs had a naming convention of initially identifying those N-linked disorders as a type 1 pattern or a type 2 pattern, and then naming them each with a letter of the alphabet. Starting with the first CDG that was identified, that was CDG1A. The second one was CDG1B, and so on. And you probably have already identified the problem with that, which was that we're now well over 100 different CDGs that are known. And hence the naming convention has changed from, for example, CDG1A to now PMM2-CDG. So PMM2 is the gene that encodes for phosphomonomutase. And so the name of the clinical disorder is PMM2-CDG, meaning the CDG associated with dysfunction of the PMM2 enzyme. Sometimes the conditions are still referred to by the old naming convention. So they're still a little bit interchangeable, but there's definitely a clear shift towards the new naming convention. So there are a couple of new diagnostic techniques and techniques that are used even for surveillance in addition to diagnosis. And so one technique is to take that transferrin and put it in a mass spec machine. And again, you're looking for a charge to weight ratio, but you have higher sensitivity with the mass spec versus the isoelectric focusing. For example, this is a type 1, this is actually a mixed type CDG that is represented here. And you can see the peak where you would expect the normal 4-sialic acid residues on a transferrin protein, but you also see an abnormal peak in that type 1 pattern position, meaning a 2-sialic acid residues plus the transferrin, also a transferrin with no glycan structures. But then this is, again, a mixed pattern. So you see evidence of the type 2 pattern as well, where there are 1 and 3 sialic acid residues. And this is from a paper that's demonstrating the potential efficacy of treatment with galactose for a condition called PMI-CDG. And so they're demonstrating that after treating with galactose, you get a much more normal glycosylation pattern. And so that is starting to be used as a monitoring technique in some cases where there are potential treatments available for specific CDGs. The other advantage of this mass spec technique is that you can look at a protein called ApoC3, and you can get a sense of one of the O-linked pathways in addition to that N-linked pathway that is assessed with transferrin. A newer still technique is what's called N-glycan profiling. So this is also using a mass spec machine, but adding a step of digestion. So you're digesting the N-glycans off of the protein, and the resultant smaller glycans can have a characteristic profile for specific CDGs, and this N-glycan profiling can be abnormal and can be diagnostic for CDGs that don't have characteristic findings on those other techniques that I mentioned, where you're looking at the intact combination of the protein plus the glycan. And so both of these techniques are used clinically, and these are kind of the two main biochemical approaches to identifying N-linked disorders of glycosylation. So by far the most common disorder of glycosylation is PMM2-CDG. So that's a deficiency of phosphomonomutase. I mentioned earlier that that's an enzyme that's responsible for one of the steps in really creating the kind of mannose substrate that's needed to construct N-linked glycans. And so the specific step is converting mannose 6-phosphate to mannose 1-phosphate. This causes broad disruption of N-linked glycosylation, and there are symptoms in many body systems. This is the classic disorder that does have an N-linked, type 1 N-linked CDG pattern. And this is a condition, there are a lot of novel mutations, but in the European population, particularly the Northern European population, there's one particularly prevalent mutation, the arginine at position 141. And one of the really interesting things about this mutation is that it's both the most common and it's also never seen in homozygosity. So that tells us that this is a complete loss of function. Even though it's a missense change, it results in a complete loss of function of the enzyme, and that complete loss of PMM2 enzymatic activity is not compatible with life. So those are non-viable pregnancies. So kind of knowing the mechanism of this condition, there's no proven therapies for PMM2-CDG, but understanding the mechanism does give you some ideas for potential therapies. So there are planned trials to look into whether administration of mannose 1-phosphate might be a viable treatment strategy. Also knowing that there is always, in all living individuals with PMM2-CDG, there's some residual enzyme activity, raises the question of whether like a chaperone type therapy might be beneficial. These are pictures of a few patients from a recent publication from the CDG group in Spain. There are some facial dysmorphisms that are associated with PMM2-CDG. They're not super reproducible, but certainly a couple things to point out are that patients often have upslanting palpebral fissures, fuller cheeks, a longer philtrum, larger ears, and the strabismus is almost universal for patients with PMM2-CDG. So given that we know that on one end of the spectrum, people with a pregnancy that lacked PMM2 enzymatic activity entirely would be non-viable, there's then an entire spectrum of disease from the extremely severe neonatal onset presentations all the way to much milder manifestations depending on the degree of residual enzyme activity. So of the patients that are correctly diagnosed, about 20% of diagnosed patients die in infancy. And the cause of death tends to be enteropathy leading to difficulty feeding, TPN dependence. The enteropathy is kind of compounded with the fact that there's severe proteinuria and hypoalbuminemia. And then, so for a patient then that is dependent on having an indwelling catheter for maybe both albumin infusions as well as for nutritional reasons, there are also coagulation abnormalities in both directions. So there's risk of bleeding and risk of clotting. So these can be very medically complex patients, patients that can be in the NICU for months at a time. And that combination of, of interventions, of the need for an indwelling line, that kind of chronic nephrotic syndrome picture with the coagulopathies that are associated can be fatal about 20% of the time. So if you think about the mechanisms of end-linked disorders of glycosylation, some of these manifestations start to make sense. So for example, the charge barrier in the kidneys relies on the fact that circulating proteins have a negative charge. And so those cells in the kidneys need to also have a negative charge on the cell surface in order to repel the circulating proteins and keep them in circulation and keep them out of the urine. So that mechanism, the mechanism of end-linked glycosylation kind of makes sense for why you would end up with proteinuria. The coagulation abnormalities also kind of make sense. So the coagulation cascade is this incredibly intricate interaction of circulating proteins, and those proteins have to be really finely tuned in terms of both their charge and their spatial characteristics, in terms of how they interact with one another to get that perfect balance of bleeding and clotting. And so disruption of the glycan structures on the proteins of the coagulation cascade cause problems, again, in both directions, bleeding and clotting. There are certainly neurologic manifestations in infancy. So these infants are hypotonic, they have developmental delay, and a significant fraction of them have seizures. The last thing that I'll mention from sort of a mechanistic perspective is a lot of endocrine disruption. So like the coagulation cascade, the circulating hormones, in order to fine-tune their activity, they have a really kind of nuanced interaction between the circulating hormones and the cell surface hormone receptors. And so as a result, changing those N-glycan structures can impair the normal interaction between hormone and receptor. So for kids with PMM2-CDG, that can often manifest as hyperinsulinism. For kids that have a somewhat milder set of manifestations associated with PMM2 deficiency, these kids often present with developmental delay, so it's hypotonia, but it's really ataxia, sort of out of proportion to other aspects of developmental delay. Cognition is typically moderately impacted with an IQ of 40 to 70. There are often asymptomatic liver function test abnormalities that normalize with age. And that's seen with a number of CDGs. It's an interesting phenomenon, but it seems that the hepatocytes, as they regenerate, they sort of go through a selection process. And those hepatocytes that are capable of better tolerating the glycosylation abnormality sort of win out, and over time the LFTs normalize often. These patients also can have some coagulation and endocrine abnormalities. There are often thyroid hormone or thyroid test abnormalities that don't actually result in clinical hypothyroidism. So many, if not most, of the PMM2 patients that I can think of actually flagged for abnormal thyroid on newborn screen. Some of these patients can have retinitis pigmentosa, and as I mentioned earlier, strabismus is almost universal. For those that are even more mildly affected, they may just have basically an isolated neurologic presentation with mild intellectual disability, some progressive ataxia. Patients can have peripheral neuropathy. Especially girls can have abnormalities of the reproductive hormones. And so the lack of spontaneous puberty can result in osteoporosis, as well as some intrinsic skeletal disease. If you look at the pathway here for generating the substrate for mannose incorporation into N-glycans, PMM2 is right here. One step up from that is mannose phosphate isomerase. So that's responsible for converting fructose 6-phosphate to mannose 6-phosphate. And as a result, you can actually get a lot of generation of mannose 6-phosphate by just supplementing with mannose. So this is one of the few, although growing, number of CDGs that are treatable with supplementation of a specific sugar. MPI-CDG has many of the same features as PMM2-CDG, but without the neurologic disease. So it's mainly the enteropathy, the liver disease, hyperinsulinemic hypoglycemia. So if you need to glycosylate proteins in order to accomplish all these physiologic functions that we've talked about, you also need to deglycosylate proteins sometimes. So this will be the first of about a half dozen or so brief mentions of specific disorders that highlight other pathways that are not N-linked glycosylation. So there is a protein responsible for, or an enzyme responsible for deglycosylation of proteins. And it's encoded by a gene called N-gly1. N-gly1 cleaves the glycans from misfolded proteins that need to be subject to degradation. And when those glycans can't be cleaved, those proteins can't be degraded as efficiently, and you end up with signaling abnormalities as well as protein aggregates that disrupt the function of cells. So this is a disorder of deglycosylation, and the features of N-gly1 include developmental delay, microcephaly. Again, we get this interesting phenomenon where the LFTs are abnormal in infancy and then they normalize over the first few years of life. And then this characteristic feature of alacrima. So these are, this is a group of patients that were published a couple years ago. They're ordered in terms of age. And when you look at the older folks at the bottom right here, you can see that they have a significant degree of chemosis or injection of the whites of the eyes, and that's because of that alacrima, and you sort of get a keratopathy related to that. So this is a disorder that was actually identified by families who got together on social media and recognized that their children all had this characteristic feature of alacrima in addition to neurologic disease. We've talked about how glycans mediate cell-cell interactions, and that is particularly important in muscle fibers. So muscle fibers all sit together in bundles, and they need to have good interactions between the individual cells to maintain the integrity of the muscle fibers. Dystroglycan is a heavily glycosylated protein that mediates that interaction. And if you lack dystroglycan or have abnormally glycosylated dystroglycan, then you get a muscular dystrophy, because as those muscles contract, they don't have the normal strength of interaction with the extracellular matrix, and those muscle fibers start to break down. So there's a whole group of muscular dystrophies called dystroglycanopathies. And this is a disorder of O-linked mannose. So if you remember, I said earlier that the O refers to this being an oxygen bond rather than a nitrogen bond. O-mannose means that mannose is the initial sugar that is attached to the protein via that bond. Those patients that have mild dystroglycanopathies have isolated limb-girdle muscular dystrophy. Those patients with more severe disease have clinical syndromes that have been termed congenital muscular dystrophy, Walker-Warburg syndrome, or muscle-eye-brain disease. And so the name muscle-eye-brain disease comes from the fact that it is a muscular dystrophy, as I mentioned, and also that the formation of the brain does not occur in the typical fashion in the absence of normal O-mannose. So this is a representation here at the bottom right of the O-mannose pathway. This would be at the bottom a dystroglycan protein. And then that green circle always represents mannose. So mannose is that initial sugar that's attached, and there's this 4-glycan structure that's the mature glycan from the O-mannose pathway. The GPI anchor disorders are a pretty rapidly growing group of disorders. They are recognizable by the genes. A lot of them start with PIG. And so there's a whole series of PIG-A, PIG-N, PIG-O, PIG-Q, et cetera. There are a number of genes that don't follow that naming convention that are still GPI anchor disorders. These photos are from a recent publication called Lessons Learned from 40 Novel PIG-A Patients. So that just came out a couple months ago. Those patients that are on the milder end of the spectrum have seizures and developmental delay. Those patients with more severely impacted enzymatic activity can have some characteristic physical features. So, for example, you can see some dysmorphisms of the ear, hypoplastic finger and toenails, and sort of tapered fingers here. One biochemical feature that is sometimes seen is elevated serum alkaline phosphatase. So if you remember, I said that the purpose of the GPI anchors is to tether a cell surface protein to the anchor that's sitting in the membrane. And so the alkaline phosphatase that would normally be bound to a cell surface is not bound to the cell surface because of the GPI anchor dysfunction, and hence it's in circulation, and as a result you can have elevations of the measured alkaline phosphatase. So one of the kind of characteristic syndromes would be severe seizures, developmental delay, microcephaly with elevated alkaline phosphatase. That would clue you in to think about the GPI anchor disorders as one possible diagnosis. This is a representation of the pathway involved in constructing the GPI anchor. So this is the anchor itself, and then the glycan is constructed with the addition of each of these sugars, glucosamine, followed by several mannose residues, and then that glycan structure is what binds to the cell surface protein. Glycosphingolipids are a group of molecules that are really critical in neurologic function. So they are involved in the cell membranes of neurons and in myelination. And so these, in general, there's just a few known glycosphingolipid synthesis disorders. They typically present as severe neurologic disorders with seizures and profound developmental delay. This pathway, just to orient you, this is the ceramide, that lipid base of these molecules, and then there's a complex series of steps by which sugars are added to create the glycan head structure for the ceramide that gives it the characteristics of the different gangliosides, like GM1 ganglioside that probably a lot of people are familiar with based on the storage disorders. But in this case, we're talking about the synthesis disorders for this group of molecules. So there are a couple of enzymatic steps here that have been identified as causing human disease. And again, those are typically pretty severe neurologic diseases. To highlight another group of disorders of glycosylation, there are a number of things, a number of genes that are necessary for normal Golgi function. So the Golgi has a characteristic both anterograde and retrograde transport. There are genes that are responsible for maintaining the pH of the Golgi, genes that are responsible for tethering the Golgi, and basically most things that can go wrong with Golgi function will result in a dysfunction of multiple glycosylation pathways. So this was a case that I was involved with. This was a patient that presented as basically a congenital muscular dystrophy. So had neurologic involvement with brain atrophy, had liver involvement. So this is an electron microscopy showing steatosis of the liver, MRI of the muscles showing muscle breakdown consistent with congenital muscular dystrophy. And then this is what's called dystroglycan staining on immunohistochemistry of a muscle biopsy. And you can see the control is on the left here. There's uniform staining of dystroglycan around each muscle fiber. And in our patient, the muscle fibers are dystrophic, and there's very inconsistent staining of dystroglycan. At the bottom here, you can see this is a assay of Golgi transit, and in control cells, this dyed substance has been fully transited through the Golgi and out of the cell by 2 hours. And in our patient cells, the Golgi transit is dysfunctional. It takes much longer. And as a result, the glycosylation that's happening in the Golgi is tuned to the time of the Golgi transit. So if you disrupt the timing of the Golgi transit, there's abnormal glycosylation, both in the N-linked and the O-linked glycosylation pathways that occur in the Golgi. The last pathway that I want to mention for CDGs is the O-xylosylation pathway. So xylose in the glycan alphabet is represented by the star symbol. So what you're looking at here in the upper right is this is the core protein of a glycosaminoglycan. At the top is heparan sulfate, and then dermatan and chondroitin sulfate, which you all, I think, know well based on the discussion that you had recently of the storage disorders associated with those mucopolysaccharides. All of those mucopolysaccharides have the same first 4 sugars. So that's called the tetrasaccharide linkage region. And so there are 4 different enzymes that are responsible for attaching those specific sugars to make that linker region. And it's probably not a surprise that if that mucopolysaccharide synthesis process is not functioning, the result is a severe connective tissue disorder. So these are some photos of patients with B3-GALT6. So that's a galactosyl transferase. In red here, that attaches the third sugar in this glycan. And so if you are unable to efficiently synthesize mucopolysaccharides, you have both developmental delay, but in addition, you get cutis laxa, severe scoliosis, kind of a Ehlers-Danlos type presentation. So this has been termed clinically progeroid type Ehlers-Danlos syndrome. So I hope that that gave you a sense of sort of the very broad spectrum of disorders of glycosylation. I will maybe stop here for just a couple minutes and take questions about CDGs, and then we can move on to mitochondrial diseases. Any questions about CDGs?
So nothing's come through on the chat. If anybody has any questions, they can speak up and unmute or send a question to me if your microphone's not working. We'll give it a minute or so, and then if nothing comes up, then we can move on and save things to the end.
All right. Given that there are no pre-existing questions, I'm just going to move on. So I'll spend the next 40 minutes or so giving you, again, the kind of very broadest introduction to mitochondrial diseases before Dr. Van Hove dives deeply into the biochemistry in a couple weeks. So I want to start by sort of helping you to conceptualize mitochondrial diseases. So there's a number of things that sort of get muddled when we're talking about mitochondrial diseases. It's important, of course, to differentiate those diseases that are mediated by mtDNA mutations versus those that are mediated by nuclear DNA mutations. It's also really critical to be clear if we're talking about a clinical syndrome versus a genetic etiology. So I want to start by talking about Leigh syndrome. So Leigh syndrome is a clinical descriptor, and that refers to a patient with some neurologic symptoms, typically developmental delay or neurologic regression, plus typical findings on brain MRI, plus genetic or biochemical evidence of mitochondrial disease. So it's those three things together make for a clinical diagnosis of Leigh syndrome. This MRI on the right is a really kind of classic MRI appearance for Leigh syndrome. So this is a FLAIR image. It's axial. It's through the basal ganglia, and so you can see this kind of classic appearance of striatal basal ganglia injury in the setting of typically with an acute illness or fasting or some other kind of physiologic stressor that provokes the acute injury. It's almost always bilateral, and it's almost always the kind of deep structures of the brain that lead to that characteristic appearance of Leigh syndrome. There are at least 75 different, and I'm certain there are more than that now, genetic etiologies that have been identified to cause Leigh syndrome. Those are pretty evenly split between nuclear DNA and mitochondrial DNA. And the characteristic clinical course of Leigh syndrome is that the initial injury, the initial symptoms happen early in life, and it tends to have a progressive course. Although the rate of progression can vary dramatically depending on the specifics of the genetic etiology. And I'll talk more about that when we talk about some specific genes. So the Leigh-like brain MRI appearance is symmetrical T2 hyperintensity, or often now we're looking at diffusion weighted imaging in the very acute period. So high T2 signal in the brainstem, the periaqueductal gray specifically within the brainstem, in the midbrain, in the medulla, and in the basal ganglia, as well as the dentate nuclei of the cerebellum. The neurologic symptoms that result are dependent on the area of the brain that's injured. So, for example, and the MRI can help us to guide management. So, those patients that have brainstem lesions are at high risk of central apnea. So, having that brain MRI appearance, knowing that there are lesions in the brainstem, would lead us to maybe get sleep studies regularly and to be very aware of the potential for central apnea for that patient. Whereas a patient with injuries that are isolated to the basal ganglia might have much more in the way of dystonia, and their management would be focused on controlling the symptoms of dystonia. If you do have access to MR spectroscopy, then there's often elevated lactate at the time of the acute injury. Classically, but not always, the onset of symptoms is in the first 2 years of life. Our kind of definition of Leigh syndrome has been getting somewhat more inclusive over time to include patients that don't have the inevitably progressive and fatal disease. So we have some patients who have been very stable, even for decades after their acute initial injury. As I mentioned, the symptoms that result are directly related to the area of the brain that is damaged. So that can include dystonia, ataxia, hyperreflexia, central apnea, dysphagia, and difficulty feeding. And characteristically, these brain injuries happen in the setting, again, of some kind of physiologic stressor, often the first serious respiratory viral illness that a patient has. So this graph looks exactly like the CDG graph in terms of the number of identified genetic etiologies of Leigh syndrome. Just like with CDGs, there's been an explosion in recent years with the broad uptake of next-gen sequencing, whole exome, whole genome. Earlier on, most of the patients who were genetically diagnosed had mtDNA variants, and later on, these have all been nuclear DNA variants that have been discovered in the exome and genome era. So just to give you a sense of the different pathways that, that can be impacted to cause the clinical picture of Leigh syndrome, that includes the pyruvate dehydrogenase complex, all 4 or all 5 of the electron transport chain and oxidative phosphorylation complexes, deficiencies of coenzyme Q10, disorders of mitochondrial DNA maintenance, mitochondrial translational disorders. And so we're gonna, again, do kind of a whirlwind tour of at least one disease from most of these pathways to give you a sense of just kind of how broad the phenotypic spectrum is and how broad the different biological mechanisms are. So the first half of this is going to be a discussion of mitochondrial DNA mutations, and the second half will be nuclear DNA mutations. So the most common single mutation that causes Leigh syndrome is a mutation at mitochondrial DNA position 8993. So most people are pretty familiar with this, but the mitochondrial DNA is a circular structure. It has about 16,000 bases. And so position 8993 is about halfway along the mitochondrial genome in a gene that encodes for ATP6, which is part of the complex 5 of the respiratory chain. So this is a really nice one to start with because it really helps us to talk about this concept of heteroplasmy. So heteroplasmy is the idea that unlike the nuclear DNA, where every cell has 2 copies of each gene, the mitochondrial DNA has many, many more copies per cell. So the mitochondrial genome is present in several thousand copies per cell, up to hundreds of thousands of copies in a few cells. And unlike the nuclear DNA, where you either have 0%, 50%, or 100% of the copies of the gene affected, you could have any percentage at all affected. So that could go from 0 to 100%. You could have 87% or 36%. And that can also vary by tissue. So some tissues can have high heteroplasmy, whereas other tissues will have low heteroplasmy. That dictates the phenotype that an individual has as a result of an mtDNA mutation. So in the case of 8993 mutations, there are two different mutations that can occur at that position. There's the T to G mutation and then the T to C mutation. The T to G mutation is more disruptive to that protein, and as a result, individuals are more symptomatic at lower levels of heteroplasmy. So folks that have less than 50% heteroplasmy are very unlikely to have any significant neurologic symptoms resulting from the 8993 T to G mutation. But as you get up to 85, 90, 95%, the likelihood of significant neurologic disease goes up to almost 100%. So an individual who is homoplasmic, which is the term for 100% of mitochondrial DNA has a particular mutation, those individuals are virtually guaranteed to have severe neurologic disease related to the presence of this mutation. The T to C mutation, by contrast, is less disruptive of that protein. So folks that have less than, say, 75% heteroplasmy for that mutation are unlikely to have any symptoms at all. And then as you get to very high levels, you're much more likely to have neurologic symptoms. The degree of heteroplasmy can vary significantly from generation to generation, and even within generations. So I certainly care for patients where one sibling has very high heteroplasmy, has severe symptoms related to an mtDNA mutation. Another sibling might have just 10% less heteroplasmy, lower heteroplasmy, and might have no symptoms at all or very mild symptoms. So it's quite striking when you start to have these big pedigrees of families that carry mtDNA mutations, just how variable the heteroplasmy is and how variable symptoms can be from family member to family member. So individuals that have moderate heteroplasmy for the 8993 mutation often have a phenotype called NARP. So that's neurogenic weakness, ataxia, and retinitis pigmentosa. Often the first symptom is ataxia and learning difficulty. That can often be coincident with a worsening of vision. So in the upper right, you can see a representation of retinitis pigmentosa. That's this darkening of the retina, worsening of peripheral vision and night vision. In the bottom right, you can see an MRI that shows cerebellar atrophy. So there's too much CSF around this cerebellum, and you can see the fovea too clearly. That tells you that this cerebellum is undergoing atrophy. And that's the genesis of the ataxia symptoms for folks with NARP resulting from moderate heteroplasmy for the 8993 mutation. You can demonstrate neuropathy using nerve conduction studies. And these folks have progressive cognitive symptoms and becomes a dementia as the decades go by, and have very slowly worsening neurologic symptoms. Sometimes folks can have hearing loss, cardiac arrhythmias, and ophthalmoplegia. Another mitochondrial condition that folks have usually heard a lot about is called MELAS. So MELAS is mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes. This MRI on the right is from a patient who is having a stroke-like episode as a result of MELAS. You can see that there's an area of the cortex that is T2 hyperintense on this T2 weighted image. You can also see that that's not in a clear vascular distribution, so it differs from a thrombotic stroke. Most patients that have the MELAS phenotype have a mutation at a position called 3243. And that's in one of the tRNAs. So the mitochondrial DNA has its own tRNAs that are part of the translational apparatus within the mitochondria for converting mtDNA into the proteins in the mitochondria. The presentations for folks with the 3243 mutation are widely variable, but a typical patient with kind of relatively high heteroplasmy might have onset in childhood with some developmental delays, maybe headaches, failure to thrive. Folks are usually quite small, so they're short-statured and have low weight. They often have exercise intolerance and weakness. In addition to these kind of the MRI appearance of the stroke-like episode, you can also have calcification of the basal ganglia. So that's a somewhat characteristic MRI appearance for some individuals with the 3243 mutation. Diabetes is quite common, as is cardiomyopathy. So both of those are quite prevalent in individuals with this mutation. Despite the stroke-like episodes kind of looming large in the description of this condition, it's a relatively small number of folks with the 3243 mutation that have those recurrent stroke-like episodes. This is on the right here, this is a representation of a muscle biopsy with a Gamori trichrome stain that highlights ragged red fibers. So that's the accumulation of mitochondria at the periphery of a muscle fiber. That's kind of a classic histopathology finding that is suggestive of mitochondrial disease. The 3243 mutation characteristically is it decreases with age in terms of the heteroplasmy in blood. And so in some individuals, especially if you're testing them later in life, they may have very low heteroplasmy in blood. That was more of a problem in prior eras of genetic testing when we didn't have as good of an ability to detect mutations at low heteroplasmy. But muscle tissue is still usually the gold standard tissue to test when you're looking for the heteroplasmy of a particular mitochondrial mutation. Luckily, we do have access to a number of other kind of non-invasive tissues now. So we'll use cheek swabs, sometimes we'll use urinary sediment, and both of those, the heteroplasmy is higher relative to blood for some of these mutations that drop out of blood with time. If you're looking at the mitochondrial electron transport chain enzymes, again, Johan will go into this in detail in a couple weeks, but for all of these mutations that impact mitochondrial translation, you see a deficiency of enzymes 1, 3, and 4. For most patients with MELAS and many other mitochondrial conditions, you can see an elevation in lactate and alanine. I always prefer to look at the alanine as opposed to the lactate because the lactate is so subject to artifactual elevations. A consistently elevated alanine, especially the ratio of alanine to other amino acids, is a much more specific indicator of a chronic lactic acidosis. So this is a nice paper that demonstrates how the heteroplasmy of the 3243 mutation changes over time in different tissues. So you can see that the heteroplasmy in blood at the lower left is dropping with age. So these are individuals between age 20 and age 80 with the 3243 mutation. And those individuals, particularly those that have higher heteroplasmy initially, as the years and decades go by, that measured heteroplasmy in blood drops considerably. In contrast, the heteroplasmy in muscle remains higher throughout life just because that's a much more stable tissue. It's less subject to the selection process that happens in hematopoietic tissues. So those blood cell precursors are constantly selecting for better mitochondrial function, and the cells that make it into circulation are those that have better mitochondrial function. So over time, the heteroplasmy drops in the blood cells. We do have some specific treatments for MELAS. So we will use arginine, citrulline, or taurine. All three of those have been shown in somewhat small studies, but appears to be a consistent signal that individuals who have recurrent stroke-like episodes will have fewer and less severe stroke-like episodes with those treatments. So many physicians will have their patients on either arginine or citrulline or taurine or combination of those to try to prevent stroke-like episodes. The physiology of the stroke-like episodes appears to be vasoconstriction, and the supplementation of arginine or citrulline can contribute to nitric oxide that can maintain vasodilation. The mechanism of the taurine is a little bit more complicated, and I'll leave that for Johan to talk about in a few weeks. In addition to Lee syndrome, NARP, and MELAS, a lot of people have heard of MRF. So MRF is an acronym that stands for myoclonic epilepsy with ragged red fibers. The vast majority of those with the clinical syndrome of MRF have a genetic etiology for that of a mutation in mitochondrial DNA at 8344, and that's a change of A to G. That also, like MELAS, or like the 3243 mutation that is in one of the tRNAs, in this case, it's the mitochondrial tRNA for lysine. And just kind of differentiating these two disorders, there's a lot of overlap, but folks with the 8344 mutation are more likely to have myoclonus, so those are sudden jerking movements, more likely to have epilepsy. And characteristically, this is a really striking and very specific finding for the 8344 mutation, is this large lipoma at the base of the neck or at the upper back. That is very specific, and you can almost do targeted testing for the 8344 mutation if you have a patient with neurologic symptoms, in particular ataxia or epilepsy, plus this lipoma in this specific location. Those folks with MELAS are more likely to have stroke-like episodes, migraines, diabetes, and cardiomyopathy, as opposed to those with MRF. The care of patients with MRF is kind of representative of the care that we provide for patients with a wide variety of mitochondrial syndromes. So there's a publication from 2013 suggesting that patients with MRF should have regular neurologic exams, echocardiograms and EKGs, ophthalmology exams, respiratory function tests, swallowing evaluation, endocrine evaluation, audiology evaluation, and then kind of neuropsychiatric testing. So that's a good representation of the sort of organ systems that we want to cover for many, if not most, patients with multisystemic mitochondrial syndromes. So the association of diabetes with the 3243 mutation is quite striking. It's there's a prevalence of diabetes of over 80% by the time folks are over 60 years old among individuals with the 3243 mutation. So that's led to this clinical syndrome that's termed maternally inherited diabetes and deafness. So that's sort of the very mildest clinical syndrome that is associated with the 3243 mutation. Those are the two symptoms that we would see at low heteroplasmy. So we see a lot of families where there are a couple individuals with diabetes and deafness, and then in a third generation or fourth generation down, there's someone who had the bad luck of ending up with high heteroplasmy, and then they might have a much more severe MELAS type presentation in a family of a lot of individuals with diabetes and deafness. The mechanism of diabetes is a little bit complicated, but it involves both insulin resistance in the peripheral tissues as well as hyposecretion of insulin. For most patients with mitochondrial diabetes, they end up on much lower doses of insulin than folks with type 1 diabetes. And we have identified an adult endocrinologist who's interested in looking for these patients with mitochondrial diabetes. So In particular, they'll look for patients that are on low doses of insulin, and they are insulin dependent, but they're on low doses of insulin, and they have deafness. But even if they don't have deafness, if they lack the antibodies that are consistent with type 1 diabetes, we have a pretty high prevalence of detecting the 3243 mutation in those individuals. We do know that a significant fraction of individuals with hearing loss have mitochondrial DNA mutations. So in particular, there's a mutation at position 1555 that is consistent or makes an individual susceptible to aminoglycoside-induced deafness. So those particular antibiotics are inhibitory of mitochondrial function, and the cochlea and the inner ear are particularly susceptible to that inhibition of mitochondrial function, and that can result in deafness in the setting of that exposure. So why is that? Why is it that there's an antibiotic that we use that is that can inhibit mitochondrial function? And it comes down to what's called the endosymbiont hypothesis. So the origin of multicellular species of eukaryotes was when a bacteria and an archaea got together. So an archaea is another whole kingdom of cells that swallowed a bacteria, and then that bacteria became an endosymbiont. So it lived within the archaea, and then that gave rise to the whole lineage of eukaryotes. But the mitochondria have retained the molecular mechanisms from that bacterial heritage, and as a result, a number of the antibiotics that work as antibiotics by inhibiting bacterial protein synthesis, they also, to some degree, inhibit mitochondrial protein synthesis. So for the example of the aminoglycoside-induced deafness, that is enough of an inhibition of mitochondrial protein synthesis to result in a phenotype of deafness. Most of the mtDNA conditions that we talked about are present in some degree of heteroplasmy. So most pathogenic mitochondrial DNA mutations are not homoplasmic, and they're not inherited from a homoplasmic parent. So one of the ways that we can interpret the significance of a mitochondrial DNA mutation is to test the mother of an individual. And often if there's a rare variant that looks somewhat benign and it's homoplasmic in the person that we're testing, we'll test mother. Mother is also homoplasmic, then we can kind of write that off as unlikely to be causative, at least of severe disease in childhood. The exception to that is Leber hereditary optic neuropathy. So there's a series of mutations, 3 mutations in particular, that are quite prevalent in folks with Leber hereditary optic neuropathy. And they are homozygous. They're present in the genes that encode complex 1 proteins within the mitochondrial DNA. And it leads to this very characteristic presentation of sudden loss of vision, like over the course of weeks. On an eye exam, and it's often asymmetrical, so one eye will go before the other, and the eye exam, the retinal exam, shows an atrophy of the optic nerve. One of the things about LHON that makes it a great thing to study is that it's not fully penetrant. So we can actually use LHON to study the environmental, the kind of genotype by environment interactions that lead to disease. So there's some really cool studies about LHON, of the effect of altitude, the effect of environmental pollutants, both low altitude making it more penetrant, environmental pollutants making it more penetrant, smoking making it more penetrant, and being male appears to be the main environmental risk factor for LHON to manifest the phenotype. The last of the canonical mitochondrial clinical syndromes is called Kearns-Sayre syndrome. Kearns-Sayre syndrome refers to individuals that have a large deletion of mitochondrial DNA. And it's important to know about this because the deletion of mitochondrial DNA is very unstable in blood tissue. And so if you're testing blood for an mtDNA deletion, you're likely to miss it. And you'll need to test, muscle is the ideal tissue, but buccal cells are also much more stable than blood. So most patients with a pathogenic mtDNA variant have inherited it from a mother that has somewhat lower heteroplasmy. So 80% of the time, mtDNA point mutations are inherited. In contrast, deletions are inherited only 5% of the time. So 95% of the time, these are de novo and not inherited. One of the characteristic findings of mitochondrial disease is this complex of symptoms that's described as CPEO, chronic progressive external ophthalmoplegia. So that's inability to abduct the eyes, and also the progressive ptosis, or lowering of the eyelids. Many of our patients with mitochondrial disease have had to have surgery to raise the eyelids so that the pupils aren't obscured by the progressive ptosis. There's a big complex of features that go along with Kearns-Sayre syndrome in childhood. So the CPEO is one of them. That tends to be a little bit later onset. But really characteristic things include hearing loss and cardiac conduction abnormalities. Diabetes, again, is quite common. Retinitis pigmentosa is quite common and fairly specific in the presence of some of these other features. CPEO as a phenotype refers to just the eye findings without the other systemic features. Pearson syndrome is the name for those folks that have very high heteroplasmy for that deletion. It is present in the blood cells. It hasn't been cleared from the blood cells, and the result is a severe transfusion-dependent anemia. Now, if you continue to transfuse the individual, then over time, that competitive process will take over. And even if there are very few, if there's very low heteroplasmy for mtDNA without the deletion, over time that will always outcompete, and then that anemia resolves. However, the other big feature of Pearson syndrome is exocrine pancreas dysfunction, resulting in malabsorption and failure to thrive. And that anemia and exocrine pancreas dysfunction is on top of all those other features of Kearns-Sayre syndrome that I mentioned. A couple of nuclear DNA mitochondrial conditions to mention. So we'll start with some of those that encode proteins of the respiratory chain. Complex 2 genes are interesting. So complex 2 is important to know about because it's exclusively encoded by nuclear genes. So if you're doing mitochondrial enzymatic studies and you find that someone has a deficiency of complexes 1, 3, and 4, but not complex 2, that would indicate to you that there's a problem with mitochondrial translation. Conversely, if there's a specific complex 2 deficiency, you would think about a pretty small number of genes. So SDH, A, B, C, and D, and a couple others that, that add on to that. The interesting thing about complex 2 mutations is that a child who has biallelic disease or recessive disease related to mutations in complex 2 genes might have a parent who has paraganglioma or pheochromocytoma, because being heterozygous for a mutation in these genes is a cancer susceptibility syndrome. So we have a couple patients who we've identified as children that have neurologic syndromes related to SDH, A, B, or C, or D. And then by identifying that child, we identify that there are multiple family members that need screening for paragangliomas and pheochromocytomas and ongoing management in a cancer predisposition syndrome clinic. I'm going to skip this one for the interest of time. SIRF1 is a good one to know about. So this is a gene that isn't actually one of the structural proteins of complex 4, but it is necessary for complex 4 assembly. So it's what's called an assembly factor. I mention it because it's one of the most common causes of Leigh syndrome. It also has kind of a characteristic phenotypic finding of hirsutism. So if you have a patient with Leigh syndrome and hirsutism, you can specifically think about SIRF1. But the other thing to know about it is that it has a somewhat milder course than some other causes of Leigh syndrome. For example, complex 1 mtDNA mutations. Those patients with Leigh syndrome, due to SIRF1, on this Kaplan-Meier plot, you see that they tend to survive longer. And we do care for a patient here who's in his late 20s with Leigh syndrome due to SIRF1, and he's been relatively stable with some mildly progressive neurologic symptoms after his initial basal ganglia injury in early childhood. I'm going to skip this one as well. So pyruvate dehydrogenase is an important group of conditions to know about. So this is a representation of the pyruvate dehydrogenase complex, which is a very large enzyme complex. Most patients with pyruvate dehydrogenase deficiency, we know of about 13 and probably a few more by now, causes of pyruvate dehydrogenase deficiency. But the vast majority of patients have mutations in this gene called PDHA1. And the important thing about it is that it's an X-linked mutation. So the importance of that is that boys and girls have very different phenotypes related to PDHA1 mutations. Boys tend to have milder mutations, so they have hypomorphic mutations, sort of going all the way back to PMM2-CDG. It's sort of analogous in that those boys that have severe mutations are non-viable. So the only boys that we meet with PDHA1 mutations have hypomorphic missense mutations. They tend to present with ataxia, hypotonia, developmental delay, may have a very significantly elevated lactate, and their lactate to pyruvate ratio is likely to be low, meaning that the pyruvate is relatively more elevated than the lactate. We can do, usually this would be as with almost anything, that would be a genetic diagnosis, but we can still do enzyme assays for pyruvate dehydrogenase. And it's important to know about because there are several potential treatments. So because pyruvate dehydrogenase is the last step that allows carbohydrates to get from glycolysis into the TCA cycle by converting it from pyruvate to acetyl-CoA, you can actually bypass that step. And for individuals that get the vast majority of their calories from fat, meaning the ketogenic diet, those folks can have both improved development, significantly improved hyperlactatemia. We have a couple patients who we've seen improvement in their brain MRI findings after starting ketogenic diet for pyruvate dehydrogenase deficiency. There's an upcoming clinical trial of a drug called dichloroacetate that's an activator of pyruvate dehydrogenase enzyme activity. Girls have, sometimes girls can have a similar presentation to boys, but other girls will have a very different presentation because they're actually mosaic for severe mutations that completely eliminate pyruvate dehydrogenase enzyme activity. So the majority of the girls that we diagnose present with structural brain malformations because of the absence of pyruvate dehydrogenase activity in utero. Interestingly, and this may be kind of something that throws people off in terms of the diagnosis is that those girls are unlikely to have significant elevations of lactate because they have enough tissues throughout their body that have good expression of pyruvate dehydrogenase enzyme activity to not have an elevation in lactate and pyruvate. The mitochondrial DNA maintenance disorders are an important category of conditions. So Alpers syndrome is caused by biallelic mutations in POLG. That's one of the most common mitochondrial conditions that we see in childhood. The important thing to know about Alpers syndrome is that in a child presenting with epilepsy, it's important to avoid valproic acid if there's any consideration of POLG as a potential diagnosis, because giving valproate for seizures can precipitate liver failure. There's a very poor prognosis for those patients with POLG that present in childhood. They tend to have progressive epilepsy, and it tends to be fatal within a few years. That's in contrast to a lot of the adults that we see with POLG. So some adults can have very late onset of symptoms into the 5th, 6th, sometimes 7th decade of life. Those are typically folks that have dominant mutations. So there's a very small number of mutations in POLG that can cause a phenotype when they're heterozygous. There are also adults that have biallelic mutations, and they can have kind of an intermediate phenotype with neuropathy, dysarthria, ophthalmoparesis. All of the late onset mitochondrial DNA maintenance conditions really classically have the findings of CPEO that I showed you earlier. I will just highlight maybe 2 more conditions here before we take a couple questions. So there's a group of disorders that result from an inability to synthesize CoQ10. So CoQ10 is a lipid molecule that is responsible for shuttling of reducing equivalents from complex 2 to complex 3, and also from complex 1 to complex 3. And when you have the inability to synthesize CoQ, that can often result in a neurologic phenotype. But patients with a little bit milder deficiency of CoQ10 synthesis can actually present with steroid-resistant nephrotic syndrome. And that can respond dramatically to treatment with high-dose CoQ10 supplementation. So this was a paper from the New England Journal in 2008. They demonstrated that a patient with steroid-resistant nephrotic syndrome due to a CoQ10 synthesis defect had complete and durable resolution of their nephrotic syndrome with CoQ10 supplementation. And we've had that experience here as well with the patient. It's quite dramatic when you're able to identify that diagnosis and treat it. I will finish up by mentioning that the dynamics of the mitochondrial membranes are really critical to mitochondrial function. So we have a lot of images of mitochondria as these little discrete beans that kind of sit in the cell and pump out energy. But that's really not the case at all. So the mitochondria really are a dynamic network. They're constantly undergoing fission and fusion to become larger and smaller and migrate to different parts of the cell and take on different functions. So there's a condition called OPA1 related optic atrophy. So we already talked about that phenotype of optic atrophy as it relates to Leber's hereditary optic neuropathy. But this is an example of another condition that can cause a very somewhat similar phenotype. And the mechanism is really interesting. So OPA1 is a protein that is responsible for mitochondrial fusion. So you can see some cells here in the bottom right that are deficient in OPA1 function, or OPA1 is inhibited, you get this very fragmented network of mitochondria. And so the ganglion cells in the eye, those very long neurons where the cell body is in the eye and then the axon goes back to the brain, these fragmented mitochondria are not able to use the molecular motors that traffic them from the cell body down the axon. So the axons of the neurons that originate in the eye are sort of starved for mitochondrial function, and they atrophy. Lastly, I'll mention that the mitochondrial membrane has some very unique characteristics. So the inner mitochondrial membrane has these this very tight folded structure, and that tight folding of the inner mitochondrial membrane allows for efficient function of the electron transport chain and efficient supercomplexing of mitochondrial enzymes. And if that structure of curvature of the mitochondrial membrane is disrupted, then you get dysfunction of the mitochondria. So that ability to create those tightly curved membranes is related to the presence of a molecule called cardiolipin. So cardiolipin is a lipid molecule that gives the inner mitochondrial membrane its unique characteristics. There's a gene called TAZ that encodes an enzyme called tafazin. This is also an X-linked condition, and tafazin is necessary for the synthesis of cardiolipin, and hence in the absence of cardiolipin, you get abnormal folding of the mitochondrial membrane. I want to draw your eyes to this biomarker called 3-methylglutatonic acid. So that's kind of a characteristic biomarker that's elevated in the group of conditions that I would call mitochondrial membranopathies. So there's sort of 10 or so conditions that disrupt the mitochondrial membrane, and many of them can have elevations of 3-methylglutatonic acid as a characteristic biomarker. The clinical features of Barth syndrome is a characteristic type of cardiomyopathy called non-compaction cardiomyopathy, as well as neutropenia and skeletal myopathy. So a boy with left ventricular non-compaction cardiomyopathy, especially if they have neutropenia, should have their 3-methylglutatonic acid checked to look for the presence of that biomarker that would indicate that the child has Barth syndrome. So I would like to end there and take a few questions. Apologies for running a little long there. And sorry for skipping over a few slides, but I'm happy to chat if anyone has questions.
That's good, Austin. We had a couple questions come through on the chat line. First, Ping Wankichote, I hope I pronounced your last name right, Ping, I'm sorry, but CDG, going back to those real quick, do you see regression? In other words, a period of normal development and then loss of skills, or is it just a chronic global developmental delay? If you could answer that quickly, and we've got a couple more.
So it depends on the specific CDG. I will answer specific to PMM2 CDG. The patients do not have regression of cognition. However, the ataxia is progressive. So they have progressive cerebellar atrophy, worsening ataxia over time, but stable intellectual disability.
Good. D. Burkhart from NIH, there's a couple questions. One is, in MELOS, and specifically in acute stroke-like episodes, when you're using arginine, can you comment on oral versus IV, and then also chronic therapy for prevention of stroke-like episodes? I think Johan may cover some of that, but if you could touch on it, that would be great.
Sure, yeah, so the regimen that has been studied for sort of as an abortive therapy for an acute seizure would be an IV arginine bolus followed by ongoing IV administration for the duration of the stroke-like episode. There have also been studies of arginine and citrulline as a preventive. So it's again, been studied both as an abortive and as a preventive medication. So IV when it's being done as an abortive therapy, but daily enteral administration as the preventive therapy.
Good. And then also from D, and I think this is a very interesting question. I'm curious to hear your opinion because I know Johan will have his, but electron transport chain enzymology, is there still a utility or added value in light of molecular and genomic testing?
That's a great question. The role has definitely changed in the genomic era. So the respiratory chain enzymology at one point was sort of a first line test in the evaluation for mitochondrial disease. I would say for me at this point, the indications are either a suspicious variant of uncertain significance on genomic testing that needs functional evaluation, or a patient with very specific phenotypic features that are suggestive of mitochondrial disease, but negative genomic testing. So those, for me, those are the two indications. So we don't do a lot, at least I don't do a lot of biopsies for enzymology in a diagnostic capacity anymore.
And then Ping also had the question, and this is a great one for clinical mitochondrial care. If you have a mom who has known heteroplasmy, how do you counsel on recurrence risk in offspring?
Yeah, it's very hard. So a mother who is known to have, let's just say 50% heteroplasmy for, say, the 8344 mutation, we would counsel that there is some likelihood of recurrence, of occurrence in a future pregnancy. It's very hard to predict what the likely heteroplasmy will be for a child. There are varying practices around the world in terms of prenatal testing for mtDNA variants. Most people, I would say, are fairly comfortable testing for the 8993 mutation specifically, because we have we know that mutation tends to be fairly uniform throughout different tissues. So we would expect that an amniocentesis would probably be somewhat representative of the heteroplasmy in the brain, which is what we really care about. The other thing about the 8993 mutation is that we have really good data about the heteroplasmy phenotype correlation. So we do feel like if we do get a certain heteroplasmy on prenatal testing, we would be able to provide some prognostic information based on that. So it's very hard. I mean, there's just a lot of uncertainty when you're counseling mothers with mtDNA variants.
Good, thank you. And then Josh Manor from Baylor, are there any clues that would point towards CDG1B to be able to treat with mannose pre-symptomatically, maybe even in utero?
I don't know about in utero. I will say it's a very rare condition. It's one that I've never personally managed, but it's important to think about based on the potential for treatment. I would say that if you're looking for specificity, the combination of enteropathy, hepatopathy, and hyperinsulinism, that would be fairly, relatively specific. That would be a early indicator to you to test, or maybe, if you very clearly had all three of those phenotypic features, maybe even empiric treatment if you were awaiting a diagnostic result.
Great. We have a couple more. One is also from Ping. In complex 2 deficiency, the paraganglioma pheochromocytoma phenomenon, are the variants you see with that similar, identical, or different to ones that you would see with Lee disease associated with complex 2?
That's a good question. So it really is, the cancer predisposition syndrome results from being heterozygous for the mutation. and then there's a second hit phenomenon within the susceptible tissue. And the mitochondrial dysfunction leads to the secretion of kind of growth factors that then results in cancer. So we definitely, I believe it's the same mutations, but it's just the difference of being constitutionally heterozygous with a second somatic mutation within susceptible tissue versus being constitutionally biallelic. So that's sort of the distinction.
Good. And the last one that's come through is actually something I was going to ask you after this if nobody else asked it. But Ali Al-Bashri from the NIH asks, is urine, and my scenario is going to be, is buccal swab a better representative sample for heteroplasmy and mito disease compared to blood? And is there any diagnostic difference in sensitivity?
Yeah, great question. So I have definitely had patients where there was a mutation that was absent from blood that was present in buccal tissue. I guess the way that I would think of it is that muscle is going to be the most sensitive, blood is going to be the least sensitive, and urine and buccal tissue are going to be intermediate. I don't know, between urine and buccal tissue, I don't have a good sense. Maybe the study's out there, but I haven't seen it myself, of which of those two non-invasive tissues is more sensitive. Certainly, we have somewhat of a hard time successfully extracting DNA from urine. So the buccal swabs are just technically more feasible. So I usually, if I'm doing a first-line test for looking for mtDNA variants, I will almost always use a buccal swab as my first pass at it. If I still had a very high suspicion of, in particular, single large deletion mediated disease, so the kind of CPEO into current SARE spectrum, then a muscle biopsy would definitely be indicated if we don't see it on buccal swab. So that's kind of how I would think of it.
How often have you tried getting urine sediment and having it either fail or succeed? I feel like the times I've gotten it, it hasn't worked out.
Yeah, I would say it's been successful less than half the time in terms of successfully extracting DNA from urine.
But buccal you've used frequently?
Yes, yeah. A buccal swab as the sample is typically my first line approach for looking for mtDNA mutations.
Great. Well, thank you very much, Austin. This was broad two topics that you approached and did an awesome job. I learned a lot myself, and on behalf of the fellows, I want to thank you. A lot of stuff coming in over the chat line to say thank you too. So any other questions, I'm sure people can email you. That your email is available on the schedule. And otherwise, tune in next week, guys. Chuck Venditti is going to be talking to us, and have a great weekend. Thanks.
Thanks for listening, everyone.